Metagenomics is, as we know, is a way to study microorganisms in their natural environment. By exploring the species richness and by investigating the functional roles of these microbial communities and what they play and the role that they play in many areas that are related to human health and human disease and in our general well-being. So in this webinar, you will learn about the upcoming OmicsLogic's Metagenomics program. And these programs, as we know, and as we have been presenting, are designed to be rewarding to students, researchers, faculties, and citizen scientists, and etc., etc. Many people who are generally interested in understanding omics analysis to several different omics data profiles. The program in general will cover main aspects of metagenomic sequencing, the analysis of 6-genus rRNA sequencing data, and the other aspects of microbiology research using bioinformatics and downstream analysis of metagenomic sequencing outputs using R and Python and using code-free platform, etc., using the Taubot bioinfo. You will be familiar in metagenomics data analysis and metagenomic sequencing generally, analysis in general, and you will be able to conduct independent projects and independent analysis, and you will be equipped to handle data from publicly available resources. Can somebody mute? Yes, thank you. Am I audible still?
Yes, sir, you're audible.
I think my screen share gone. Okay, wait a second. My screen share is creating some problems. Okay, I think I should be, you should be able to see my screen.
Yes, sir, it's visible.
That's perfect. So I was talking to you about how generally this program will equip participants like you to be able to conduct independent projects using metagenomic sequence analysis data. And you will also be able to handle and find data from publicly available resources and public domain. And you can also learn from high impact biomedical publications on the microbiome, the kind of analysis that they perform, and what are they actually referring to when they are talking about the diversity, and that also leads to dysbiosis and symbiosis of microbial population in relationship to the phenotypical characteristics of the sample or under different biological conditions with which the samples are actually recorded or referred to. So as in all of our OmicsLogic training program, we will use online sessions, we will use asynchronous sessions, we will use self-guided study materials, and we will use several different example projects that will help you get a effective understanding and good experience of understanding the concepts related to the metagenomics sequence analysis and metagenomics data analysis. So let's dive into this session with the focus on metagenomics, focusing mostly on the bioinformatics part related to the analysis of the sequencing data. So before we begin, let's see and how are the different areas with which we can relate the metagenomics data and metagenomics information to, and ask important questions about these microbial communities, and also review a couple of studies that are used to characterize microbiota from different sources. So the connection between disease and microorganisms has been established in the 1800s, and we already know that towards the end of the 19th century, we have several established laboratory methods for growing microorganisms in lab culture. Right? And these developed methods of growing these organisms in laboratory them with staining and other methods and helps us understand their characteristics and helps us understand their relationship to our diseases and pathogenicity in general, right? And these studies in the earlier stages of understanding the microorganisms helps us, or they actually provided us important information about these bacteria or these microbes and their function in isolation. But however, it was soon apparent, and we already found out that most of these microorganisms live in a complex community. Today, we know that these microorganisms, or we call them as microbiota, that make up the human microbiome, are not just unicellular organisms living alongside each other, but instead they form highly regulated, highly structural, and highly functionally organized communities that are skin attached to several different organs, and they collaborate with the organs, and they contribute to the ecological, and they contribute to the stability and well-being of the tissues and of the organism that they are found part of. So to give an estimate, the human microbiota is estimated to be around 10 to the power 13 or 14 of microbial cells. And they roughly translate into 1:1 ratio of microbial cells to human cells ratio number. And these microbes, they form distinct communities, and they are interconnected to maintain their community. The stability of these communities in their own environment is a fascinating study, and the researchers that helps us understand these stabilities and the effort that these microbes put in to maintain these stabilities are fascinating, and they give us several insights into how these stable environment relate to our well-being and our reaction to different disease states and different pathogenicity. And we already know that, and we already also see that bacteria can communicate with each other by producing, detecting, and responding to several small diffusible signal molecules. The sensing of these signal molecules are also involved in the virulence and the pathogenic potential of the bacteria, and is therefore an important factor in understanding and controlling bacterial infection in general. So inside and alongside each of these communities, there is a balance of bacteria, fungi, and viruses that control species abundance and activity. So in each of these environment, each one of these microorganisms carry important functional properties in genes that contain genomic information. Right? We can study the genomic data of these microorganisms to understand the properties of the genes and to understand the functional importance of these microorganisms in their environment. These properties and the genomic information about these microorganisms have been of a growing interest to the scientific community, and it has already resulted in over 80,000 publications in total, and it's still growing. And the majority of these studies have actually focused on intestinal microbiome, and several other areas of microbial population and studies are also coming into the research or coming into the highlight. So, however, since 1990s, microbiome studies are actually significantly advanced through several major initiatives like the metagenomics of human intestinal tract and human microbiome project, and huge studies. Several such studies have made possible through the advances in metagenomic sequencing in general. And these sequencing, which came as a result of development in high throughput sequencing efforts or next generation sequencing effort, have actually made it possible that there is an ever increasing amount or ever increasing availability of the data that can easily pour into several of the derived data, several of the derived projects and research that are fueled by the existence of such biological data. So as a result of these studies, skin, lung, gut, and even brain microbiota are starting to be getting characterized, and their relationship with our well-being and in general, their involvement in maintaining balance between general biosis of our microbial community are constantly being explored and related to. So today we know that the endogenous human microbial communities contribute to critical metabolic, critical physiological, and immunological functions. And these are all functions that include differentiation and maturations of the immune system. And sometimes they include food digestion and nutrition elements. And they also include metabolic regulation and control of the fat storage or energy storage. And they are also involved in processing and detoxification of environmental chemicals. And we also know from acne and other related studies, they are involved in maintaining skin and the mucosa barrier function. And more relations or more key connections are establishing or getting established in the development and regulation of the immune system. Inflammatory and anti-inflammatory substances are also getting actively studied on. So prevention and invasion of disease-promoting microorganisms and the general colonization resistance and general resistance of advantageous microbes to these disease-promoting microorganisms are also being actively explored. So the term metagenomics covers all of these studies and refers to both the research technique and also the research field. So the field can be defined as the genomic analysis of the microbial DNA from these environmental communities that we discussed on, and the tools that used to study these analysis enable us to understand or enable us to make population analysis of previously unknown microbes or previously unculturable or uncharacterized microbes or bacteria in general. This is very, very important in the current scenario because only around 1 to 2% of the bacteria can be cultured and grown and studied in the laboratory. And the ability to identify these microbes without a priori knowledge of what the sample contains is actually opening new doors into the research areas like ecology, like virology, microbiology, environmental science and biomedical research, etc., etc. The sequencing-based examination of the metagenome has become an extremely powerful tool for understanding their diversity and their effects in several of these research areas that I just listed on, and has become very useful tool in generating novel hypotheses that relates these metagenome to several of these research areas. So this sequencing can actually apply to any culture-free environmental population, as well as it can be applied to any general detailed extraction of microorganisms that can be characterized with or without culturing in labs, and often times in order to gain a complete understanding of these selected few microorganisms. These techniques are combined. First, a detailed or an exploratory metagenomics approach is being applied to some to culture-free sample, and then from these selected few microorganisms are actually grown in the laboratory culture to explore and to verify the hypothesis that we generate from the metagenomic sequencing or these culture-free sequencing techniques. So many of these questions are being answered, as I mentioned, using the tools such as using the very important tool that is next generation sequencing, which focuses on either sequencing the whole genome or the whole or it can focus on sequencing selected gene like 16S rRNA or 18S rRNA of the microbes, or sometimes selected portions of these genes of the microbes, right? And in performing these different sequencing methods, we will focus, or we are going to focus on answering or addressing several important, different key questions, and that will help us generate hypotheses related to the data, related to the biological condition of the samples that we have. So in general, when we look or when we discuss about these different technologies and these different methods that are used to characterize metagenomics and microbial population, we have shotgun metagenomics, and we have 16S metagenomics or amplicon sequencing methods. While the shotgun metagenomics or transcriptomics, metatranscriptomics will focus on understanding all of the genes or the whole genome of the microbes, and this can help us in analyzing, for example, the general assembly with the help of the general genome assembly of these several different microbes. We can get key insights into their functional contribution in maintaining the balance and in helping us with the processing of the metabolites. We can also prediction of the genes across to be a key factor or key player in maintaining this metabolome and their processing levels. So while shotgun metagenomics and transcriptomics will address at the functional level of the microbial population, in addition to sometimes giving us an estimate of these microbial population, more more reliable methods in estimating the abundance level of these taxonomical level can be performed using 16S amplicon metagenomics, right? This 16S sequencing is a technique that is widely used to study a microbiome directly in its environment, and this will focus on either, as I already mentioned, 16S or 18S ribosomal RNA that has to be amplified using PCR amplification method before getting quantified and matched onto the database of known 16S rRNA sequences. So these sequences contain hypervariable regions and variable regions that provide species-specific signature sequences that are extremely useful and are used to identify bacteria and their taxonomic ranks. How are they generally performed? So they are actually, in addition to sequencing, these PCR amplified reads or PCR amplified shared products of these 16S rRNA gene, we pool them, or we generally cluster them in group of closely related individuals as defined by their taxonomic identity or taxonomic or sequential identity or sequential similarities, right? These consensus sequences are actually defined as operational taxonomic units, and their abundance is actually characterized and quantified for different samples. Yes, Ram Kumar, OTUs are no longer in use as we have ASVs, because ASVs can be a general term that can take anywhere between 95% of the sequence identity to 99%. OTUs are restricted in the amount of identity that are used to define how two sequences are related to each other. That is why. Okay, it's not the case that it is not generally in use. It is still in use, but in order to have a very generic application of the sequential identity and their relationship to identifying key taxonomic ranks, we are referring to ASVs. Okay. All right. So they can be classified or they can be clustered using general clustering approaches that will cluster these reads based on the percentage identity level that we are defining while defining the amount of the taxonomic units that we want to characterize. So we were discussing that these taxonomic units are actually taxonomic organization in terms of kingdom, phylum, class, genus, etc., etc. So these OTUs are the one that are identified as specific species or genus or phyla of bacteria, and they are the one that are that will get characterized or that will get quantified among different samples that we have. So once this quantification is performed, as I mentioned, they are identified to be part of these specific ranks. Using special taxonomic information for domains of bacteria, archaea, eukarya, and etc. etc. Right? It is primarily based on the phylogenies of the subunits of rRNA, and thank you, Ramakumar. And yeah, subunits of the 6 genus or 8 genus rRNA and their relationship to the taxonomic ranks that we already discussed about. So in addition to this, we also have virome, which refers to one part of microbiome that constitutes all of the organisms, or that constitutes all of the viruses that might inhabit us, right? So characterizing the virome of mammalian samples are our samples, and their relationship to causing disease is also extremely important, because sometimes these viruses can be carrier for several other they are also implicated in being carrier to oncology related issues and oncological implications, and several tumor progression are also being implicated to these abundance levels of specific viruses.